This is a guide of how you can perform the ELISA test for the ANA9 kit. It will give you visually all the information you need to do the test, including the use of the Terrace software. All kit boxes are labeled with the content of the kit, the expiration date, and the kit's lot number. Remove all reagents and plates from the kit box and allow them to come to room temperature, which is 18 to 25 degrees Celsius, for 30 minutes. Each kit contains a data sheet with the unit values for the calibrator and control ranges. These units and ranges vary with each new lot. Prepare all necessary equipment to perform the test. They are listed below. Label each dilution tube with the calibrator, control, or sample ID for the purpose of keeping track of the location of each tube. Calibrators and controls should be loaded according to the following plate maps. Notice how calibrators and controls are absent from the second plate. All calibrators and controls should be vortexed prior to being added to a dilution tube. In a separate dilution rack, load 125 microliters of each pre-diluted calibrator into its respective dilution tube in the following order: blank, single-strand DNA, double-strand DNA, SM, RNP-SM, SSA, SSB, chromatin, SCL70, and centromere. For the remaining tubes, which contain controls and samples, add 1.1 milliliters of specimen diluent. Note, because the largest pipette can hold only up to 1 milliliter, 550 microliters of diluent should be added to each tube twice. Using a new tip each time, pipette 11 microliters of controls and samples into their corresponding dilution tube. After adding each sample to its corresponding dilution tube, move the dilution tube over one place to indicate that the sample dilution is completed. This serves as a precautionary measurement to save your spot and avoid confusion when diluting further samples. Cap all samples firmly after adding specimens. Slowly invert the dilution tube rack 5 times in order to mix the samples. Note, as a means of ensuring adequate mixture of the samples, watch as the fluid moves through the dilution tubes. Using a pair of scissors, carefully loosen and remove the caps from each sample tube. Set a timer for 30 minutes. Using the 12-channel pipette, carefully draw up 100 microliters of each calibrator. Observe each pipette tip and ensure the fluid levels are equal throughout. Load the calibrators into their corresponding wells in the first row of the microwell plate. Using the multi-channel pipette, carefully draw up 100 microliters of the remaining samples, which are the controls and patient specimens. Load the controls and patient specimens to their corresponding wells. Note, to avoid contamination or scraping antigens off the wells, never touch the inside of the wells. Once loading is complete, start the 30-minute timer. While the plates are incubating, prepare the wash buffer. Using a graduated cylinder, add 900 milliliters of deionized water and 100 milliliters of 10x concentrated wash buffer. Following the preparation of the 1x wash buffer, take the label from the concentrated 10x wash buffer and place it on the newly prepared buffer. Initial and label the buffer with the date it was created. Once the timer sounds, take the plate to a sink designated for biohazard disposal and decant it by flicking the plate in a swift downward motion. Pour 1x wash solution into a clean reservoir. Set the multi-step pipette to number 5. This will load 250 microliters with each press of the lever. Insert 8 new pipette tips firmly. With the tips in the wash buffer, depress the multi-step pipette plunger to load the wash solution. Carefully pipette 250 microliters of 1x wash into each column. Lay the pipette down on the bench with the tips facing down and out. This will prevent any fluid backing up into the pipette and contaminating it. Decant the wash into the sink. Repeat this wash procedure 2 additional times for each plate for a total of 3 washes. Firmly and carefully tap each plate upside down into a paper towel. This is performed to remove as much excess wash as possible. Pour conjugate into the reservoir to get ready to load. Use new pipette tips to load 100 microliters of conjugate to each well. Start a 30-minute timer. The leftover conjugate may be poured back into the conjugate bottle. When the 30-minute timer sounds, wash the plate with the same technique used for the first wash cycle. Wash each plate 3 times. Use a new reservoir for the addition of the chromogen. A full plate of wells will require 10 milliliters of chromogen. For 2 plates of wells, add 20 milliliters of chromogen. Plan accordingly. Excess chromogen may not be poured back into the original bottle. Set a timer for 15 minutes. Start the timer as you begin to load chromogen into the first column of wells. Pipette 100 microliters of chromogen from a reagent reservoir into all of the wells. Do not touch the edge of the wells with the pipette tips to avoid contamination. Never pour chromogen back into the reagent bottle. This will contaminate the rest of the chromogen. Discard any leftover chromogen. Add stop reagent to a new reservoir before the timer expires. When the timer sounds, pipette 100 microliters of stop reagent into all of the wells. Tap the side of the plates gently to mix the wells. The rest of the stop reagent may be poured back into the reagent bottle. Place the developed plate into an ELISA reader for analysis. To read a new plate on the Terrace software, select File, Reading, New. Select ANA9C from the list of assays. Select Reader, Read and Collect. The reader will take 75 to 85 seconds to read the plate. This is an example of absorbance measurements displayed on Terrace software for the ANA9 assay. Wells A1 through A10 represent calibrators for each respective antigen. Wells B1 through B10 and C1 through C10 represent positive and negative controls respectively. All rows that follow represent patient samples. After measuring the absorbance values, select the Sample IDs tab. Select Edit On. Enter the sample IDs from the assay in the same order the samples were loaded into the microplate wells. Select Save once all sample IDs have been entered. Following the absorbance reading, save the test by selecting Save. A new screen will immediately pop up, and in the dialog box, type in the name of the test that is run, followed by the date and the initials of the individual who ran the test. Please see the example. Select OK to save the assay. This concludes the presentation provided by Theratest Laboratories in reference to performing the ANA9 ELISA assay, as well as a demonstration of test result storage with our proprietary software, Terrace. If further clarification or assistance is required, feel free to contact our technical support department at 1-800-441-0771.